Thanks to the organizing committee for providing me the chance to present my lecture. I am Abhinav Mishra from Research Scholar from the Department of Physics, Dindayal Upadhyaya, Gorakhpur University, and under the joint supervision of Professor Subrinath Tiwari and Dr. Dipen Sharma. My research topic is DFT and molecular docking studies of a naturally occurring anti-cancer drug, Ellipticine. Basically, Ellipticine is a naturally anti-cancer drug which was demonstrated by the elliptica tree in which we investigated. Now, next to the introduction part, firstly in 1959, isolation and structure determination of 6H-pyrido 4,3-B carbazole alkaloids from the leaves of Ocarina elliptica label, which was carried out. Ellipticine has basically a planar structure and plays a significant role in the treating kidney cancer, anti-cancer drug, phytobiologic lymphoma, open breast cancer, metastatic and brain tumors. Basically, natural agents are proving to be an important source of novel inhibitors and have the potential for development into selective anti-cancer agents. Lee Picard et al. 1973 synthesized the potential anti-cancer drug for anti-cancerous activities using the interaction with DNA. These workers used their results to correlate the drug activity and intercalability. As a result, there is a major research effort which seeks to gain deeper insight into the molecular basis of interactions between small molecules and DNA with special emphasis on the site, mode, sequence, and structural specificity of their binding mechanisms. In order to understand better the molecular basis of these important and complex interactions, it is often advantageous to use simplified model systems. The study of DNA, RNA, small molecules interactions provides well-defined models that allow general principles for biomolecular complex formation to be established and help the drug discovery. The present paper basically embodies in silico investigation of the physicochemical and electronic structure properties, map surface, homo-lumo energy, global activity descriptors of the ellipticine drug using ab initio DFT method and basically the spectroscopic and in IR and UV spectra. Under using the for interaction, protein interaction, basically ligand and protein interaction, we use the molecular docking technique. Using molecular docking technique, elucidation of binding mechanism of ellipticine with 3 isomerase transcriptases taken from the protein data bank with PDB ID, 1 DID, 2 IPI, and 1 XIG has been accomplished for discovering the pharmacological properties of the drug. Now the objectives of my research work is based on the crystallographic data. The molecular structure of the ellipticine molecule will be designed using suitable visualization software, optimized structure and molecular parameters will be obtained. Basically, in my research work, we use the Gaussian software with and Gauss view software. Electronic parameters and global activity descriptors will be calculated in order to understand the toxicity and chemical reactivity of the ellipticine drug. A spectroscopic analysis of the ellipticine drug will be carried out by computing IR and visual spectra. To check the binding of protein ligand complex, molecular docking will be performed. This will provide the binding sites of the ligand with protein receptors. This is the theoretical method. Basically we use the B3-lip and DFT method, in which DFT method before we use the Hartree-Fock method because of the major complications in Hartree-Fock method. So we use the DFT method for the present calculations. This is the density functional theory equations which represent the basis function basically, and this is the electronic parameters which we calculate the global activity descriptors, basically the ionization potential, electronegativity and electronegativity or global hardness, global softness and electrophilicity index with the help of HOMO energy and LUMO energy, and we also determine the HOMO energy and LUMO energy gap. This is the pura whole iteration process is examined in this pura whole theoretical method. This is the initial guess, calculate the effective potential and solve the Cohen-Sham equation, evaluate the electron density of the total energy, and we will check the converged or not converged. If not converged, then whole process will be start, and till the process complete, then whole the process is converged. Result and discussion. This is the optimized geometry of the calculation of the Gaussian. We optimized the structure, any drug, any biomolecules. This is the optimized geometry of the my DSC. We use the DFT method, V3-lip method, and the basis set 6311 GDP basis set. This is the frontier orbital analysis. The HOMO energy characterizes electron donating ability of a molecule, while LUMO energy determines the ability to accept an electron. Therefore, higher values of HOMO energy indicates better tendency towards the donation of electron. The energy gap between the HOMO and LUMO is very important in determining the chemical reactivity of the molecule. The high value of the energy gap indicates that the molecule shows high chemical stability, while a low HOMO-LUMO gap means small excitation energy to be manifold of the excited state. For the Ellipticine, HOMO energy gaps come out to be 3.821 electron volt, which makes it chemically more stable and suitable for electro-optical applications. Moderate value of frontier orbital energy gap also implies its interaction due to the charge transfer eventually taking place within the molecule. HOMO-LUMO orbital of Ellipticine are shown in the next figure. This is the pictorial view of the frontier orbitals of the Ellipticine. This is the upper surface. We use the HOMO figures, and the lower surface we use we see the HOMO figure. HOMO energy is the minus 5.459 electron volt, and the LUMO energy is minus 1.638 electron volt, and the forbidden energy gap is 3.821 electron volt. With the help of these two data, HOMO energy and LUMO energy, we calculate the calculation of the global reactivity descriptors of the Ellipticine drug. This is the calculated data of the HOMO energy, LUMO energy, energy band gap, electronegativity, electron affinity, ionization potential, and global hardness and global sharpness with the help of the above formula. This is the spectral analysis. This is the FTIR spectra. Infrared spectra, we use the simulation method in IR spectra. This is the pictorial diagram of the infrared spectra of the Ellipticine. This is the bibracial assignments of the Ellipticine drug. This in which we assign the different frequency pay, different frequencies pay assignments we calculate. And last with the interaction between the drug and ligand, drug and protein receptors with the interaction. Where for molecular docking, we use the Swiss dock web server, and for visualization of their results, we use the Chimera software. For molecular exploring and binding of a drug with receptor, thereby estimating the effectiveness of the drug, molecular docking is used. Thus, docking has great importance in the field of drug design. In this process, as possible conformation of the ligand and their corresponding energies are computed. The avoid sampling bias whole docking process accomplished by the Swiss dock
Mr. Mishra, conclude within 2 minutes. Your 8 minutes is over.
Okay, sir. The Ellipticine drug shows anti-cancer activity, and hence the docking study of the drug with anti-cancer reverse transcriptase with PDB-ID 1DID 2IPI and work XIG has been performed. This is the best binding pose with binding energy value of minus 8.01 kilocalorie per mole between protein residue LYS 148 and drug LIG 1S10 having distance of 2.692 angstrom is depicted in figure 4. The corresponding FF score is minus 19 to 2.419 kilocalorie per mole. Another preferred site for strong binding energy value of minus 7.69 kilocalorie per mole with FF score minus 920 0.75 kilocalorie per mole has been investigated between GLU38 and GLIG H13 at a distance of 1.865 angstrom. Similar molecular docking is observed for protein PDB-ID 2IPI and 1XIG. Thus, the judging results suggest that the drug Ellipticine exhibits strong binding affinity with the 1XIG receptor as compared to 1DID and 2IPI protein receptor. This is the pictorial diagram of the protein transcriptase protein ID 1DID. This is the ligand size LIG S10. This is the receptor size LYS, and this is the smallest distance between the best binding pose of the this interaction is distance with 2.692 angstrom. This is the another protein receptor 2IPI with ligand interaction. This is the another 1XIG and with ligand in and compared to 1XIG is better than the 1DID and 2IPI. Conclusion: the optimized geometrical parameters like bond length and bond angles and vibrational mode show a good agreement with the corresponding experimental data. In addition, the relationship between the reactivity and electrophilicity have been discussed to access the utility of global reactivity descriptors. Ellipticine configuration possesses higher electronegativity. Molecular docking simulation of protein ligand complex elucidates that ellipticine strongly binds to protein receptor, namely 1XIG as compared to other isomerase transcriptases. Thank you, sir.
Sir, my name is Mr. Roomul Mushtaq. I'm doing my PhD under the supervision of Dr. Muhammad Zubair Ansari. Department of Physics, National Institute of Technology, Srinagar. I'm here to present my presentation, which is titled as Structural, Optical, and Morphological Properties of Solvothermally Synthesized CZTS Nanomaterials in the International Conference on Composite Materials for Environment Protection and Remediation. This is the outline of my presentation. First, I will be introducing this CZTS, its synthesis, its basic characterizations like XRD diffraction, Raman spectroscopy, optical analysis, and morphological and composition analysis. The use of solar power along with the other renewable sources is now among the most plentiful and eco-friendly ways to generate power. The numerous benefits of silicon led to the dominance of solar power market. Solar cells based on silicon does not appear to have much more room to future owing to this material's low absorption coefficient. So an alternate to silicon having high absorption coefficient, that material like cadmium, tellurium, CIGS, etc. But the problem with these materials like cadmium, tellurium, selenium, etc., these were non-toxic, and the materials like indium, they were scarcity of in the Earth's crust. In that run, CZTS has been chosen, in which the elements like copper, zinc, tin, and sulfur are present, which are earth abundant and non-toxic. The abundance in the Earth's crust is like we have copper, it is 0.0068% in the Earth's crust. Similarly, zinc, tin, and sulfur. Coming to the structure of this, it has got the tetragonal structure, in which each atom like copper, zinc, and tin are tetragonally bonded with the 4 atoms of sulfur. Each sulfur atom shares 2 tetrahedrons. Why we are choosing this material is that whole lies in its property. If you look at the solar irradiation spectrum, it is irradiance plotted against the wavelength. We see there is a tremendous amount of energy per unit area per unit time available in the visible region. So we must choose a material which has high absorption coefficient and whose band gap lies in that wavelength region, like between 350 to 750 nanometers. And this CZTS material has this property that we can tune its band gap around 1.5, which is the standard value of band gap of this material. And if we look at the table where we have energy production and consumption yearly, if we see the solar flux, the solar energy that is available in the Earth's crust, it is 3850,000 exajoules around. If we compare this with the electricity production, it is 70 exajoule. It is a huge amount of energy. There is a huge amount of energy available in the Earth's crust that is directly irradiated from the solar. So we must use the materials which has high absorption coefficient and has band gap which lies in the visible region, so that we can properly convert that amount of energy into some usable form like photovoltaics, etc. I have chosen this solvothermal method for the synthesis of this CZTS material. The precursors of copper, zinc, tin are taken in a beaker and put in the hot plate, and the magistrator with the hot plate. The solution is transferred to autoclave. After that, it is placed in the oven for 24 hours, and then at a temperature of 200 degrees Celsius. Then after cooling, it was centrifuged and drying, and finally we got that CZTS nanoparticles. These are the basic characterization. One of that is X-ray diffraction. I obtained the peaks XRD of that material, and I have compared my XRD with the standard available in JCPDS number 260575, and in the table I have compared the data to theta values and D spacing of my material, and it is exactly the same as obtained in the standard values. I have calculated the crystallite size using Sharra equation, and that comes out to be around 10.1 nanometer. After further analyzed this diffraction data using reticle refinement, and I obtained the latest parameters which are quite similar to those of which I obtained from the JCPDS data and observed by from the XRD X-ray diffraction data. For this structure, I took the Raman spectra. We can see that the high intensity peak is 332 centimeter inverse, which is quite good as I compared it with the previous data or previous literature available. That this peak must lie between 331 centimeter inverse to 338 centimeter inverse. This peak arises due to vibration of sulfur atoms, corresponds to the phase of CZTS nanoparticles with crisp structure. There are two more peaks, 228 centimeter and 362. When I compared this data with the JCP with this previous available, I found that this also corresponds to the CZTS and nanomaterials. After doing that, I performed the optical absorption spectroscopy. We can see that this material absorbs a good spectrum in the visible range. We can see that from 500 nanometer to 700 nanometer of wavelength is absorbed by the material. This shows that this material can be used for various applications in that visible region. Like we can perform some photo degradation of dyes. We can use it for this hydrogen production, PEC water splitting hydrogen production. And this material has also the property that it can be used in supercapacitor applications. After that, I plotted alpha H nu versus H nu. I got the
Mr.
band
Mishra,
gap.
sorry to interrupt. Please conclude within 3 minutes.
Okay, sir. Okay, I have 2 more slides there.
Okay.
I found the band gap 1.68 electron volts. This is the FESM EDX data. Since this material has got the problem that if we took the ratio of the precursors, it does not come out similar or exactly similar that what is obtained in the EDX data. But this material exactly nearly the ratio of the material is 2:1:1:4, which is quite good. And in the micrograph, the polygon nanoparticles are very clearly visible, which are uniformly distributed throughout the sample. This is the conclusion. I have synthesized this material in the structural analysis. I have calculated the crystallite size using the Scherer equation. I find the crystal size 10 nanometer and UV DRS. I found that band gap of the material comes out to 168 electron volt. And from the EDX data, that the ratio of copper, zinc, and tin sulfur are quite good and are 2:1:4. And FESM shows the formation of polygon nanometer with some sort of agglomerated morphology. Thank you very much, sir.
So myself, Nishita Sharma. I'm a research scholar in the Department of Chemistry, Chaudhary Bansilal University, Bhiwani. My paper ID is 265 and my topic is Insights into Adsorptive and Efficient Removal of Rose Bengal Dye from Wastewater by Biomass-based Adsorbent. Basically, I am working on the dye removal by using biomass-based adsorbents. Introduction. As we all know that water is very precious for the life. It is the basic necessity for the life, but the availability of fresh water is the limited in amount, and it is getting polluted by a number of the contaminants. And in those contaminants, dyes are the most most specific. And here is a report where the water stress by the country is given for the 2040, and in which we can see that by 2040 India will be in the high risk of the water stress. It's about 40 to 80%, and major portion of the world will be under the water stress. Therefore, water reuse or the water decontamination or remediation is the most important need, the most important step that we should convey. I am focusing on the toxicity of dyes. On worldwide, about 8 by 10 raised to power 5 metric tons of more than 10,000 various synthetic dyes are assembled annually, which are being used in dyeing and other industries to provide variant colors. And these dyes are a major threat to the pollution in the form of colored water being ejected from industries and discharged into water bodies. Therefore, it is very sensitive issue to treat the dye-loaded water efficiently to make it suitable for use. Therefore, I am focusing on the biomass-based adsorbents. Further, there are a number of the contaminants that are divided into conventional contaminants and emerging contaminants. Conventional contaminants are dyes and heavy metals, whereas emerging contaminants are pharmaceutical waste, agrochemicals, personal care products, oil, phenols, etc. Here, by the report of CPCB 2021, it was noted that there are about 390 gross polluting GPI industries, and among them 317 industries are those that are textile industries, and textile industries में dyes are the major portion of that. After that, methods of removal of the contaminants are chemical methods, biological methods, and physical methods. Chemical methods contains pantone reagent, ozonation, oxidation, UV irradiation, electrochemical destruction. Biological methods contain bacteria, aerobic or anaerobic fungi, enzyme degradation, or algae. Physical methods contain adsorption, ultrafiltration, membrane filtration, ion exchange, reverse osmosis, nanofiltration. Among all these methods, I have chosen the adsorption because of its advantages over other methods. These are the high efficiency, ease of design, low cost, less harmful secondary pollutants, accessibility of a wide range of adsorbents, facile regeneration of the adsorbents. And various biomass-based composites are used for the adsorption process, such as the orange peel, coffee beans, and coconut husk, and other. They are used in either in the raw form or are modified in other forms. They are dried, grinded, and sieved, then modified with chemical or thermal methods, or used as a adsorbent. After that, in this work, my work contained the preparation of the adsorbent. I have basically used the Sacramunja plant stem. Sacramunja, that is called Sarkanda in our literature. Sacramunja plant stem were peeled off and was washed repeatedly with distilled water for the removal of impurities. The material was dried off in an oven and then grounded in an electric grinder. The material was sieved through 120 basis mesh size to reduce its particle size and to increase its efficiency to remove the dyes. Thus, the prepared adsorbent was used for the removal of Rose Bengal dyes. Further, the characterization of prepared adsorbent. The prepared adsorbent was characterized by the FSM to study its surface morphology. It was further FKR was used to know about the functional groups that are on the prepared adsorbent. XRD was performed to know about the crystal planes and crystal structure of the material. In the characterization, we found that in FSM we show that there are a large number of the pores on the surface of the adsorbent that are responsible for the dye adsorption. In FTIR, there were the peaks of the specific groups, like at a peak was found at 2900, that was due to CS2 bending. A peak was found at 3300, that was due to OH stretching peaks, and a peak was also found on 1720 that responds to C double bond O groups. And in XRD, we found that there were the two major peaks, one at the 21.6 degree, that was a sharp peak that denotes the crystalline nature, whereas a broad band was found at the 15.6 that represents the amorphous region, and that was that represents the surface that represents the heterogeneous surface of the adsorbent. For the batch adsorption experiments, to optimize the various conditions like effect of pH, effect of dosage, effect of concentration, all these were studied, and pH of the dye was adjusted by using the HCl or the NaOH solution. For adsorption kinetics, the batch adsorption study was performed in 100 ml conical flask by taking 0.01 gram of adsorbent in 10 ml of solution of 100 ppm dye at 30 degrees Celsius for time interval of 1 to 30 minutes. For isotherm studies, adsorbent doses of 0.01 gram per 10 ml of dye solution of varying concentration from 50 to 200 milligram per liter was taken, and the mixture was stirred for the 10 minutes at 30 degrees Celsius. Further thermodynamic studies were done the studies include the adsorbent dosage of 0.01 gram per 10 ml of the dye solution. The concentration was taken as the 100 ppm with a contact time of 10 minutes, and the temperature was varied from 30 to 60 degrees Celsius. The percentage amount was calculated by this formula: Ci minus E divided by Ci into 100. Q is the maximum adsorption capacity. That was calculated by the Ci minus Ce into V by M. V volume is in the liter and mass is in the gram. QT, the adsorption capacity of dyes at a particular time was calculated by this formula. Further, Results and discussion. Effect of pH. The first of all, the effect of pH was studied. To study the effect of the pH over the percentage removal of Rose Bengal dye, as Rose Bengal is an anionic dye, then we found out that as we decrease the pH, the percentage removal got increased. When we varied from the pH from the 2 to 12, it was noted that with the increase in pH, the removal decreases, and it was when we show its interrelation with the pH PZC, we found that that this type of behavior depends upon the pH PZC, as the pH PZC of the Sacramunja was about the 6.6, and when the pH is below the pH PZC, then the surface will carry the positive charge, and it will increase the removal of the negative anionic dyes. Therefore, the maximum removal was obtained at the pH 2. It showed the maximum removal due to electrostatic attractions between negatively charged adsorbent surface and cationic surface of the anionic surface of the Rose Bengal dye. After that, effect of dosage was studied. It was found out that as we increase the dosage, the percentage removal got increased because the surface area get increased. After that, adsorption kinetics, it was observed. We have checked all the models. We have checked the experimental data with all the models like pseudo first order, pseudo second order, interparticle diffusion model, and Ellipticine model. Among all them, on the basis of the R scale value, we found out that pseudo second order model was the most suitable model. And here, the QE calculated for pseudo second order is 53.39 milligram per gram. That was in good precision with a theoretical value of QE 52.87. Further, adsorption isotherm was studied. There are different models like Langmuir model, Friendly's model, Tampin model, DR model. On the basis of that, on the basis of experimental data, all the models were checked, and Langmuir model was the best suited model with the R scale value of 0.99. The Langmuir model suggests that the monolayer nature of the dye adsorption and the value of Q max was 66.62 milligram per gram. After the thermodynamic study was evaluated, delta G node is equal to minus RT ln Kc. Kc is the equilibrium constant. A graph of ln Kc versus 1 by T was plotted, and it was found out that the delta H node was 24.95.
Miss Sharma, you may conclude within 2 minutes.
Sir. That's my last slide.
Okay.
And the delta H node value was 24.95. The positive value of delta H node suggests that the adsorption process is endothermic in the nature. And delta S node value of 70.74 suggests that the degree of disorder gets modified because of the enhanced movement of the dye. And delta G node, the negative value of delta G node with the temperature demonstrate about the feasibility of the adsorption process. After the regeneration of the used adsorbent was studied with the acetone and hot water, it was found out that the material gets regenerated up to 5 cycles, which shows a high compatibility in the commercial field. Conclusion and future perspective. In short, I worked with a low-cost, eco-friendly, as well as efficient adsorbent from Rose Sacramunja for the removal of Rose Bengal dye. Effect of pH, effect of concentration, effect of doses were all evaluated. Adsorption kinetics was evaluated, and the best fitted model was the pseudo second order model, and the best isotherm model was the Langmuir model, which suggests a monolayer adsorption. Further, the thermodynamic study revealed the spontaneity as well as the endothermic nature of the reaction. The regeneration study of the adsorbent up to 5 cycles make it competent for the industrial use. My future perspective for the work will point upon the simultaneous removal of diverse contaminants like dyes, heavy metals, pesticides, and many more by using the prepared adsorbent. These are my references. Thank you.
Good evening, everyone. My name is Anjali, and I'm currently working under the supervision of Dr. Amitabh Acharya at CSIR IHPT. Today, I'm here to give an oral presentation on the topic bio-inspired carbon dots as potential nanozymes for combating glioblastomal hypoxia. For better understanding, we need to understand some keywords present in the whole title. For example, bio-inspired, what are nanozymes, and what is hypoxia or glioblastomal hypoxia? For the bio-inspired, it means taking inspiration from a biological entity, be it a structure or a process. Nanozymes, whereas nanozymes are nanomaterials which have inherent enzyme-like properties. And hypoxia is a condition where the tissues are not having proper O2 supply. Moving ahead, firstly, we need to understand what is hypoxia and how its prevalence is there in the glioblastoma. Hypoxia is a condition of insufficient amount of oxygen available to the tissues. The hypoxia condition arises because tumors are known to outgrow their blood supply quickly, leaving parts of the tumor tissue with regions of low oxygen concentration as compared to the healthy tissue. Hypoxia is also known to create drug resistance drug resistance while treatment, and this also decreases the success rate of the conventional therapies. Hypoxia in the tumor microenvironment is associated with some hallmarks, that is angiogenesis, that is the generation of new blood vessels to provide proper blood supply to the growing tumor. Immunosuppression, that is the decreased immune system activation. Metastasis, drug resistance, acidosis. How glioblastoma and hypoxia are related is further I am about to tell. Glioblastoma multiforme is type of a brain cancer which have the highest percentage of hypoxia, that is around 50 to 70%. Moving ahead, glioblastoma to summarize what is glioblastoma and its key point. Firstly, glioblastoma is a type of central nervous system cancer, and it is most common and aggressive form of primary brain cancer. It develops from the astrocytes, that are star-shaped cells that provide protection from blood diseases and provide nutrition to the growing brain. Globally, over 241,000 people die each year with glioblastoma. 5-year survival rate is even less than 4%, and it even goes down in case of older patients. Oxygen level in the brain tumor, including glioblastoma, can be around 1 to 1.25%. Further, the hypoxia creates a major hurdle in the treatment of the glioblastoma. Thus, more new strategies are required to relieve the hypoxia in case of the glioblastoma. One such therapy or technology that is coming up is nanozymes. Firstly, what are nanozymes? Nanozymes are engineered nanomaterials which has the ability to mimic the biological enzymes. Nanozymes can act as enzyme mimics. They can disrupt the key pathways involved in the drug resistance and generate oxygen in situ. When they are combined with other therapy methods, that is in the combination therapy, they provide better results. Nanozymes are being heavily explored for oxidoreductases, transferases, hydrolysis classes of the conventional enzyme. Nanozymes play a crucial role in scavenging ROS in ROS cascade by mimicking antioxidant enzymes such as superoxide dismutase and catalase to produce less toxic products, whereas the oxidase starts the whole ROS cascade. In light of these, we have prepared carbon dots and conjugated carbon dots, which can directly target the glioblastoma, cross the blood-brain barrier because of their low size, and they can directly target the glioblastoma and can act as a nanozyme. Here we have synthesized carbon dots using Vitex nigundo plant leaf extract. Then the whole extract was hydrothermally reacted, and then the purified solution was dried powder to obtain the carbon dots, abbreviated as PCD. And then this PCD was further conjugated with PEG and para-aminosalicylic acid to obtain a conjugated carbon dots. Here is the schematic representation of the carbon dot synthesis. Moving ahead, we have carried out the characterization of the carbon dots using different methods, that is UV-vis spectroscopy, zeta potential, FTIR spectroscopy. In case of the UV-vis spectroscopy, we have obtained a peak around 280 nanometer for the peak carbon dots and the conjugated carbon dots, which signifies the pi to pi star transition. Whereas we have also obtained a peak around 340 nanometer that signifies the N to pi star transition, as well as suggests the internal nitrogen doping because of the use of the plant extract. Further, we carried out the zeta potential studies, in that case we have obtained the carbon dots and the conjugated carbon dots are having negative zeta potential, indicating that the surface charge on the carbon dots is having negatively charged functional groups present on the surface. Further, through the FTIR, we confirmed the conjugation of the PEG and the para-aminosalicylic acid, which has been conjugated using EDC-NHS coupling, giving the indication of the amide bond presence. Further, we have also checked the inherent fluorescence property of the synthesized carbon dots using the fluorescence spectroscopy studies. Here we have used two solvents, that is water and 50% ethanol. In both cases, we have obtained an excitation emission profile of 430-515 nanometer. With the change in the fluorescence intensity of carbon dots were observed upon change of the solvent. Further, we have carried out TEM and TEM studies to characterize the carbon dots and the conjugated carbon dots. In case of the carbon dots, we have obtained a mean size of 7.60 nanometer. And in the EDX pattern shows the abundant presence of carbon atoms. The SED pattern suggests the amorphous nature of the synthesized carbon dots. And in case of this conjugated carbon dots, there is an increase in size, suggesting the mean size is approximately around 8.14 nanometer, with increased abundance of carbon and nitrogen atoms. And SED pattern suggests the amorphous nature of the conjugated carbon dots also.
Miss Nisha, you may conclude within 2 minutes.
Yes, sir. For the conclusion and the future perspective part, it is from what for summarizing the whole presentation, we can say that the acidic tumor microenvironment is generated due to accumulation of various reactive oxygen species, and also hypoxia exacerbates the tumor resistance in the tumor microenvironment. So in order to combat this, we have synthesized carbon dots to target glioblastoma and relieve hypoxia through green synthesis method, and also characterize the synthesized carbon dots and conjugated carbon dots by various spectroscopic methods like fluorescence spectroscopy, UV spectroscopy, FTIR, and zeta potential. For my future work plan consists of checking the interaction of different metal ions with the conjugated and bare carbon dots, carry out with different biochemical assays to assess the antioxidation activity of the carbon dots, and to evaluate the enzyme mimicking activity of the developed carbon dots for oxidoreductase class of enzyme, along with exploration for the different classes. Last but not the least, I would like to acknowledge my supervisor and director, CSIR IHPT, and of course, Research Plateau and GVM College for giving me this wonderful opportunity for presenting my work here. Thank you.
Myself, Sarita, and my paper topic is title is Adsorption Behavior of Dyes onto Pennisetum glaucum in Wastewater Effluents. Content: Introduction, toxicity of dyes, methods for removal of dyes, material and experimental studies, results and discussion, conclusion and perspective of future. Introduction. As we know, the global outlook of water pollution revealed that water treatment is necessary to the removal of toxic pollutants, clean water security and consumption. As water is constantly polluted with various types of pollutants, like conventional and emerging contaminants that are highly toxic, and some compounds like heavy metals, organic compounds, pharmaceutical waste, or are toxic even at extremely low concentrations. Although various types of water treatment methods are widely available. Our next is toxicity of dyes. As we know that the dyes disturb the whole ecosystem. These disturb the fertility of soil. Dyes absorb more light, so interrupt the photosynthesis activity and affect the plant growth. These are carcinogenic and mutagenic, causes the hazardous disease. In plants, these disturb water. These disturb the COD and BOD percentage, increase acidic and basic toxicity, and imbalance the physiochemical and biological characteristics of water. Toxicity to feces, reduction in photosynthesis activity of aquatic plant, increases pathogen number. There are various methods for the wastewater remediations, like the physical methods, chemical methods, and biological methods. In physical methods, adsorptions, membrane filtration, ion exchange, reverse osmosis, and non-filtration methods are involved. And in chemical methods, ozonation, coagulation, fluctuations, oxidation with NaOCl, electrochemical destructions methods are involved. And in biological methods, the enzyme degradation method, algae degradation, and bacteria aerobic and anaerobic methods are used. In my topic here, we have used the adsorption method over all these methods due to the following advantages, like high efficiency, simplicity of design, low cost, less harmful secondary pollutants, and a wide range of adsorbents, easily availability of the adsorbents, and facile regeneration of the adsorbents. My scheme of research paper is that the agriculture waste-based adsorbent is used, like the Pennisetum glaucum, which was collected, cutting, grinding, and bowl milling. The Pennisetum glaucum material was collected from the nearby region, and stems were obtained, bowl milling them, and a powder was obtained. So, scheme is contaminated water was taken, and in that the bioadsorbent was added, and we get the treated water. Results and discussion. Characterization of the adsorbent. After preparing the adsorbent, the characterization was done like for the surface morphology and functional groups. The Pennisetum glaucum adsorbent was analyzed with the FCM and FTR spectroscopy. XRD was performed on a X-ray diffractometer, while the thermal and changes in enthalpy were analyzed with the TA instrument. Motors make DSC. Batch adsorption studies. For performing the batch adsorption studies to investigate the effect of various parameters like pH, dosage, concentration, temperature, and contact time, we have taken the fixed adsorbent dosage in 10 ml of dye water. After that, the solution was stirred for 10 minutes, and the final concentration of dye solution was measured. Percentage removal was calculated. Effect of dosage and the pH. The adsorptive behavior of the raw Pennisetum glaucum for the removal of carrageenan dye was studied between pH 2 to 11, and adsorbent dosage was carried to 0.1. We observed that increasing the adsorbent amount, the percentage dye removal was increased. The temperature effect and contact time was the kinetic and thermodynamic studies were done, and we obtained that the kinetic pseudo second order model was best fitted, and the adsorption isotherm have the exothermic nature. The concentration effect of dye revealed that the Langer adsorption isotherm was followed. After all that, the regeneration studies were performed, and the adsorbent give the good regeneration efficiency up to 5 adsorption-desorption cycles. Conclusion is that the Pennisetum adsorbent was very effective for the removal of hazardous dye, and experimental data was well fitted with pseudo second order kinetic and thermodynamic studies, revealed the spontaneous and exothermic adsorptions, and the Pennisetum glaucum showed significant adsorption efficiency up to 5 adsorption-desorption cycles. And the desorbed dye were recovered, making the whole process eco-friendly. Thank you, sir.